All right, so good morning and good evening, everyone. So thank you so much for joining us for today's Immunozoom seminar. It's really my great pleasure to welcome today's speaker, also my colleague, Dr. Grégoire Altan-Bonnet from National Cancer Institute, NIH. A very brief introduction about Grégoire. So Grégoire actually got his bachelor degree in both mathematics and physics in France. And then he continued to complete his physics training in master also in Lyon, France. So after he completed his master's study, he pursued his PhD still in physics in Rockefeller, New York by 2000. So after he completed his PhD training, he decided to utilize his physics background to actually learn immunology, which I think is quite inspiring and then quite a brave. So he completed his postdoc training in immunology in NIH, NIAID to be specific. And then from 2005, Grégoire actually started his independent research program have the double appointment both in Cornell and Memorial Sloan Kettering in New York. So all the way up to associate member, Grégoire actually moved to NIH by 2016 to join a National Cancer Institute. And then just last year, 2021, he becomes senior investigator in laboratory of integrative cancer immunology in NCI. So Grégoire's actually been, as I said, been trained as statistical physics and nonlinear dynamics and in immunology, which his lab tried to address the interplay between the roboticness and the variability of self and non-self discrimination in the immunology, in immune system. They also focus on developing quantitative models of lymphocytes communications by cytokines. And currently, his lab focused on the multicellular coordination of immune responses against tumor and pathogenic infections. They in particular interested in developing quantitative models in integration of signal transduction, gene regulation, cytokine communication, cell differentiation, and proliferation. And actually, Grégoire's lab actually developed really fascinating computational models, which actually strongly interface with different experimental efforts. So the very long-term goal of his lab is to help to develop tailored immune therapies. The particular area his lab focus or interested is including immunology, system biology, computational modeling, machine learning, and then biological physics. I think his work is absolutely refreshing and eye-opening by using machine learning in immunology, which I personally think which is really inspiring. So with all these achievements, Grégoire actually got a different award and honors, including James Robinson Faculty Chair in Memorial Sloan Kettering. And then also he's appointed as Statman Investigator in NIH. Research Highlight Award from 2018. And then last year, he got NCI Director Award for Merit. So the most impressive thing for me is Grégoire's lab so far has been training over 40 different trainees, which are all very active and then highly achieved in different fields, including academia and the industry. So I'm very much looking forward to your talk, Grégoire. Really exciting to have you being here with us. Please go ahead.
Thanks, Joan. That's my pleasure to be here too. So good morning, good evening, depending where you are. Can you see the slide?
Yeah. Well, I mean, it's kind of a double. Yeah, now it's good. Yeah.
Very good.
Yeah, perfect.
So thanks for the kind of introduction, Joan. And again, no, that's my pleasure. And the beauty of these Zoom meetings that we are meeting people from all over the world. So I look forward to discussion. Don't hesitate to interrupt and ask questions in the middle. This is meant to be very interactive. So what I'm going to tell you about today is mostly a recent study we did where, sorry, I'm going to hide some controls. Maybe you don't see them, I don't know. But a study we did to try to develop a model of immune response. And like Chuan mentioned, we're very interested in kind of merging machine learning and immunology to really try to not only use the tools from machine learning to better tackle the complexity of immune response, but vice versa, we can really use the immune system as a kind of a complex system where we can develop new tools for machine learning. So this kind of back and forth between more computer science and more immunology gives this kind of analogy, and this is a bit for Chuan, and we call the immune system kind of a liquid brain, and it's going to become very clear as we go along to really make it explicit what's happening. So I'm based at the NIH, at the NCI in Bethesda, Maryland, on the east coast of the USA. And you must be familiar with the work of Steve Rosenberg and colleagues, but there has been a classical protocol developed for cancer immunotherapy at the NIH and beyond since then, where people try to harvest the leukocytes from a metastasis of a patient, right, from a tumor which has developed, dissociate this tissue, and then try to collect the leukocyte, expand them with a lot of interleukin-2, and then infuse them back inside the patient with some conditioning, like chemotherapy, radiation to make space. But the idea is that if we have enough T cells which are specific for these tumors, and if we can expand them and differentiate them, maybe we would have really a very nice cancer immunotherapy because they would be very specific for the tumors. And indeed, so this is a very recent paper from the Rosenberg group, where they are looking at the metastasis from breast cancer. And they did this treatment with adoptive cell therapy. And indeed, they got some success where they could really get a regression of the primary tumors and also of the metastasis in these patients. So there have been some very striking success in that context. But at the same time, there are also very striking failures. And I would say, as an immunologist, this diversity of response is kind of a challenge because we really have to understand, in one way, can we kind of tilt the balance of the immune system towards fighting the tumors without getting too much adverse events? How can we understand the balance between fighting the tumors and leaving the self alone is really kind of a big challenge, in my sense, in immunology these days. And I'm going to show you a little bit what we do in that context to really try to develop some models where we will understand better what triggers an immune response, what triggers T cells, and to really understand the balance between self and non-self and the way cells get activated. So maybe for this crowd, it's going to be kind of obvious, but I really always like to start with something very graphic, which is not from my lab, which is from the Janelia Research Lab across the river. And this is the first light sheet microscopy where we are looking at T cells in red and in a tumor in blue. And you see that this interaction is extremely dynamic, right? So it's very possible for T cells to go and recognize the tumors and interact with it in a very tight manner, and eventually, hopefully, kill the tumor cells. But again, I like to start by this image because you're going to see a lot of flow cytometry, but inside this system, there is something very dynamic that we need to understand. And we think that by studying the dynamics and by looking at the way T cells recognize the tumors, we might be able to really build a better immunotherapy. Now, when you think about it, this is a neuron, right? But when we look at the T cells come with their receptors, B cells, whatever cells in the immune system is going to come with a bunch of receptors, for antigens, for cytokines, for costimulatory signal. And every time you look at a cell which gets some input from the outside and then does something with this input and decide a response, you can think of them as kind of a neuron, right? There is kind of an input-output relationship what we need to decipher. And what people have done in the case of neurons, we can do as well in the context of leukocytes to really understand the way they get activated. And again, in the context of discussing the immune system as a liquid brain, I want to draw some parallels between the immune system and neuronal systems. You can think of them as a kind of collection of cells which are all communicating with each other. They are like sending some signal to their neighbors, and then hopefully making a collective decisions and making a functional response at the level of the tissue. In the brain or in the immune system, there's always a very large diversity of cell types. We are very familiar with T cells, B cells, dendritic cells, macrophages. Then, of course, subtypes of these different cells. Of course, in the context of neurons, you have exactly the same thing with different neurons, with different function, different type of receptors. And at the functional level, when we look at the brain, when we look at the immune system, there is this ability to classify what's happening in terms of input signals and to build memories, right? So again, we are really trying to conceptualize the immune system as a collection of cells which takes a bunch of signals and make a collective decisions. And understanding how this collective decision is being made is really a way to really move forward immunotherapy and hopefully be able to build better response. So there's a lot of similarities between the immune system and the neural system. At the same time, there are very fundamental differences, right? Obviously, the immune system doesn't have a fixed geometry, right? All the interaction between cells are not like neurons with like synapses, which are kind of hardwired in the network. In the case of the immune system, we're dealing with very fluid interactions. Cells are moving, cells are finding new partners and developing new interactions. On top of that, B cells, T cells come endowed with somatic mutations. So there's a huge diversity in the way they are being wired in terms of their receptors. And we estimate that there are 10 to the 9 T cells in the context of humans, or a lot of diversity in the way they are wired. More importantly, in terms of dynamic response, these are not static cells like neurons. They can really expand and contract, right? So we can really start from very few cells recognizing the tumors, and they're going to expand by a factor of 10 to the 6, 10 to the 7. So it's very dynamic and very exponential in the way they respond. We need that because we need to be able to match what the virus is doing or what the bacteria is doing. But at the same time, this means that this is a modality which maybe doesn't exist at a fundamental level for neurons. And again, one key aspect of the immune system is that it really performs self, non-self discrimination at the functional level. So again, some similarities, some differences. I hope it generates a conversation today, and you tell me what you think about it, if it helps you think of the immune system. So at the fundamental level, we are studying self, non-self discrimination in my group, right? So you have to think of dendritic cells or tumors or any cells in the body coming with the MHC, presenting a repertoire of all the peptides inside the cells or inside the environment. So at steady state, at homeostasis, you're going to see these cells decorated with a lot of peptides, self-peptides. And hopefully, if the T cells come about, it's not going to get activated. But then once in a while, you get neoantigens because you're dealing with a tumor which has mutated. You deal with a virus or a bacteria which has infected the tissue of the antigen-presenting cells. And suddenly, you're going to see these new peptides popping up on the surface of the antigen-presenting cells of the tumors. And the T cell which come about and scan their surface has to be able to discriminate between self and non-self to really trigger a good immune response. So at the fundamental level, we are really studying this issue of self, non-self to really understand what we need to be able to activate T cells in a very efficient manner and try to build a model of the way this decision is being made. And I've been studying that in collaboration with Paul Francois at McGill University for many, many years. I'm just giving you here a few numbers just to strike some very specific points in terms of T cell activations, just to show you how challenging it is to think about T cells getting activated. Obviously, we know that T cells are very specific, right? So we know that the difference between the self ligands and the non-self ligands at the biophysical level might be just a difference of binding, which is maybe a threefold in terms of off rate. So very tiny differences at the biophysical level. At the same time, T cell activation can be very fast. We know that you can see an immune response within seconds of T cells interacting with tumors. And it's at the same time very sensitive in the sense that you need very few ligands to activate the T cells. So if you take these three numbers at once, and if you think of the way you're going to build a model of T cell activation, it's very tempting, for example, to make the cell very specific. But usually when people build models of specificity, it takes a lot of time to come about. Or maybe it's not very sensitive. It takes a lot of ligands to really make a discrimination. When you try to reconcile these three aspects of T cell activation, this is very challenging because you need a system which is both very fast, very sensitive, and very specific. And this is really something very striking about T cells and something which has been fascinating us for many years now. Now, it's very important to understand this complex machinery because we know that this complex machinery can do many, many different things. So this is a data taken from a paper from Christina Lesdie's group at Sloan Kettering, where we are profiling CD8 T cells in the context of different mouse model, different model of infections, so classical SCMV infection. And then they did single-cell RNA-seq on these CD8 T cells at different time points in the different contexts. And what you see that, like, for few transcription factors that you might care about, like FOXO1, let's say, or TCF7 and so on, there are very different patterns inside these T cells, right? So from one given infection, from one type of immune response, you're able to expand the repertoire of T cells in such a way that we can do very different things in that context. So this is a very interesting paper if you want to have really a kind of atlas of all what T cells can do. And now if you move in the context of cancer immunotherapy, this is some data from the Sebastian and Migona's group in Paris, where they harvested CD8 T cells from the primary lung tumors of patients. And then they did single-cell RNA-seq and repertoire analysis. Again, they see a lot of diversity in the way these CD8 T cells and CD4 T cells will differentiate inside the tumor of the patients. So some of them seem to be cytotoxic, some of them seem to be terminally differentiated, some of them look exhausted, some of them are cycling, right? So within the same tissue, the same T cells seems to respond very differently, and really you have different classes of response. And I'm showing you this paper because, and there have been similar paper from Phil Bradley's group and Paul Thomas at the University of Washington and Cathy Wu at Dana-Farber, because what they did in these papers is that they overlaid the single-cell RNA-seq and the way these T cells are being wired in terms of phenotype with the clonality in terms of TCR. And what they found, which is a result which I think is very striking and which is really kind of very controversial for the field, is that it looks like at every T cells, every clone with a given TCR, it's not like they can really do all these different phenotypes. They seem to cluster in a given phenotype. So these pink clones seem to all become cytotoxic. This blue clone seems to be mostly all terminally differentiated. So it looks like each clone comes with a given TCR, it comes with a given antigen in a way, the way they get activated. It seems to really direct the way they get activated and really kind of channel the activation into very specific phenotypes. So we're very interested in trying to understand that and building models of the way these T cells get activated in the context of the tumors. Now, if you go back to very fundamental level, we know a lot about the signaling machinery of a T cell. So this is a cartoon taken from a review by Ed Palmer. You look at the receptors, it has all the associated chain. As soon as you engage the peptide MHC, you're going to start triggering some phosphorylation. You're going to start recruiting a lot of adapters and kinase and phosphatase. So we know a lot about the components associated with the way T cells get ticked and activated, but we don't really have a good model to go from the peptide MHC to the way T cells get activated. And what I'm going to show you today is kind of a method that we have developed based on machine learning, which enables us to take T cells, to look at the way they get activated, and try to build from the ground up a model of the way T cells get activated in such a way that we can really understand better the immunology of T cells in that context. So again, don't hesitate to stop me and ask me questions. I want it to be as interactive as possible. But again, we are really going back to a very fundamental question. If you give me a peptide MHC, I hope one day to be able to give you a model which is really going to predict the way T cells are going to respond, whether they recognize it as self, whether they're going to recognize it as non-self, and all the different peptides in between in such a way that we can really understand different classes of T cell activation. So today, again, I showed you a little bit of about T cell activation. I'm going to show you a model that we have developed to understand how self, non-self discrimination is being wired. And then very quickly, I'm going to move to what you was mentioning in the introduction, which is really that we developed a robotic platform to really try to build these models automatically from some machine learning. And then we show you a little bit of modeling and how we can use this model to learn new stuff in immunology. So historically, we have been understanding the way T cells discriminate between different ligands, so between self and non-self, with a very simple biochemical scheme. So historically, people looked at it as kind of kinetic profiling. So you have to think about the receptor binding the peptide MHC and starting a phosphorylation cascade with multiple steps. And every time you debind, you have to restart at 0. So this is called a kinetic profiling because in a sense, you have all these steps where you phosphorylate the receptors and you have a chance to kind of proofread the quality of a TCR peptide MHC interaction. So what we have in this kind of model is that we have only partial discrimination. If we compare red ligands, we put different quantity of the red ligands on the x-axis, and we look at the number of fully phosphorylated receptors in our model, we see that we have a very nice scaling in log-log scale. But then when we look at the different ligand, which is maybe more like the self, you're going to have exactly the same scaling. And simply by having a big quantity of bad ligands, you can look exactly like a good quantity of small quantity of good ligands. So this model was not enough. And again, with Paul François, we have been developing some models where we add a negative feedback, which has been well documented from the biochemistry, right? We think that there is a phosphatase called SHIP1 of PTPN6. And what the system does is that as soon as you start engaging the receptors, the first thing that you're going to do is to really activate a negative feedback. And what this negative feedback does is that it's really going to slow down the way you're going to make the decision for the T cells. And it's going to really forbid a big quantity of bad ligands to get activated because it activates a lot of negative feedback. So I'm going to refer you to these two theoretical reviews where you can look at a lot of the details in terms of biochemistry, in terms of the math. But at some fundamental level, we think that the key components to build a good T cells is to have this kind of kinetic profiling scheme where the better you bind, the higher you have the chance to activate the T cells. But at the same time, you're going to have a very proximal negative feedback, which is going to compete with that pathway, which is going to kind of strain and block a big quantity of self-ligands from activating the system. And it's really this competition between the positive signal and the negative signal, which really builds the way T cells get activated. So again, we understand it at the fundamental level. I'm going to skip this math for the sake of brevity. And I'm going to move to really the inspiration that a lot of what we do. When we built the model, we understood that all these steps, all these intermediate steps, which correspond to different stage of activation of the receptor. So maybe 1 tyrosine phosphorylated, 2 tyrosine phosphorylation, 20 tyrosine phosphorylation on the TCR. If you can read these different patterns of phosphorylation, you are kind of building a neuron. So you are building a system which is going to have very different states, different level of phosphorylation on the receptors, and that might trigger different type of immune response. So indeed, we looked at some old papers from Ron Germain, who was my postdoc advisor. So this is a paper from Ito and Germain in 1997, where they did a very simple assay. They measured how much cytokine is being secreted after 24 hours for TCR transgenic with different quantity of peptide. And what you see is that depending on the cytokine you measure, you're going to have different input-output relationship. So for example, if you look at interferon gamma, you need a tiny quantity of ligands to really be able to trigger these T cells. And if you want to make IL-3, you need a much larger quantity of T cells. So think about these different cytokines as kind of output, which might be reading different station, different state of a TCR, maybe different state of activation of the T cells. And in a way, you can go from, again, a ligand binding to a receptor on the surface of T cells. And this seems to project into many different patterns of inflammation. And maybe we have a chance to really learn the way they decide by looking at these different cytokines. So this is a paper from Paul François in a physics journal. If you want to learn more about the math behind that, it's very interesting patterns. So again, at the fundamental level, we are really thinking about T cells as a neuron, or in a way as a kind of machine learning machine, right? So you take a T cells, it comes with a TCR, it's scanning the surface of the tumors, scanning the surface of the dendritic cells, and trying to detect the patterns of peptide MHC. So all these peptide MHC are engaging the TCR. This is going to activate different signaling pathways. And these signaling pathways are going to activate different gene and different transcriptional response, different cytokines, maybe some proliferation, maybe some killing, and so on and so forth. So at fundamental level, if you like to read more computer science and machine learning, this really maps into a very nice set of algorithms where you start from kind of an antigen recognition step. Then there is something called encoding, where you're going to kind of take all these signals and bring it into a bottleneck and kind of make a very simple representation of what's happening at the level of the surface. And then you have kind of the decoding, where you're going to project into some output and some cytokine expansion. So really, if you're familiar with the computer science field, a lot of people are developing this kind of framework to really build intelligent machine which can take different signals and make smart decisions about them. And we are really going to use this algorithm to learn about T cell signaling. Now, what we wanted to do is to really be able to use this kind of algorithms to really build a model of T-cell activation. But to do so, you need a lot of data, right? If you go to a machine learning paper, you're going to see that most of the time, the condition to really train a good algorithm is to have a lot of data, right? So the log that we have is that in the case of immune system, we can generate a lot of data fairly quickly, right? It's fairly easy to collect some T cell from a mouse model, from different tissues, or even get clinical samples, or even get make some T cells from peripheral blood of healthy donors. And then you can collect different tumors, primary tumors or cell lines. You can have different type of antigen-presenting cells. It's fairly easy to start to assemble in an 96-well format many copies of the same immunoresponse, right? And the game changer for us that when I moved to the NCI in 2016, we started to build this robot which enables us to do that on a big scale, right? So it's not very different compared to what we used to do at the bench, assembling a 96-well format and in vitro and then collecting things by hand. But of course, because we built a robot in the lab, first, we can keep working during the pandemic, right? The robot can keep doing experiments even though we are not really in the lab. And on top of that, we can start to do time kinetics. We can start to multiplex things. We can collect different type of tissues, different type of antigens, and so on and so forth, and generate a lot of data very quickly. So if you come to the lab, these days, you're going to see this machine. In fact, I hear it taking a time point as we are talking. So this is a video that I took a few months ago where I'm showing you typically what's happening. We have this incubator on the right, which really has 40 plates, 384 plates. At every time point, it gets the robot picks up a plate from the incubator, remove the lid, picks up some tips, and everything you would do at the bench, like collecting supernatant, preparing single-cell RNA-seq, staining the cells, fixing them for flow cytometry. Well, you can program the robot to do that for you. And again, the beauty is that one, it can take time points at 2 a.m. It's not going to be complaining. And two, you can really start to really multiplex things, right? You can take very tiny amount of T cell, tiny amount of neurons, tiny amount of cardiac cells, if you care about this type of tissues, and then mix them together and try to model what you think is happening inside the organism. And here in this particular case, we're just collecting supernatant. We wanted to see what type of cytokines is being produced. So again, it's not really rocket science, right? It's really a simple robot that we custom design for our own purpose, but it really enables us to generate a lot of data very quickly. And if you're interested in developing this kind of technology, don't hesitate to contact us. We have a lot of code and things that we can share. So again, at some level, this is the classical assay that you would see in any immunology lab. You can collect a T cell, for example, from a T1 TCR transgenics. You can make some APCs, bone marrow derived dendritic cells from a V6 wild type mouse, and then mix them with different quantity of peptides, different number of T cells, different number of dendritic cells, and so on and so forth. And then you program the robot to go in there and collect supernatant or cells at different time points. And all the measurements that we do are based on flow cytometry right now. So we don't do much single cell RNA-seq just for budget reasons. But what we have is the ability to really measure 40 cytokines very quickly using these cytokine bead arrays. We have a spectral flow cytometer, which enables us to measure 35 markers typically. We can collect 18 time points, and we can program the robot to go there 18 times over like 1 week. And fairly quickly, you're going to get this big matrix of data where at every time point, you're going to have one cell where you know exactly its state of differentiation and how much cytokine was in the supernatant, and a big matrix of data. Now, if you have this big matrix of data, you can start doing machine learning. And indeed, we're trying to do a lot of single cell analysis right now. But just for today, I'm just going to show you what we did with just cytokines. And I'm going to start with this kind of panel of CAR T cells. So these are CAR T cells which have been prepared in collaboration with Naomi Taylor's lab at the NCI. So they are like anti-CD19 CAR T cells which recognize CD19 on B-cell lymphomas. And they were prepared from 6 healthy donors. And again, mixed with different effector to target ratios. And what we see is that as a function of time, right, on the x-axis, we are looking at the different time point. On the y-axis, different concentration from these supernatants. And we look at 10 different cytokines, interferon gamma to TNF alpha, and whatever you can put on this cytokine build array. And I'm just showing you that to show you how dynamic and how rich the kinetics can be. Like if you look at IL-4, for example, it ramps up very quickly, and then it gets consumed very quickly. And if you look at IP10, there's going to be a long time delay, and then the culture is going to start to react. And if you look at interferon gamma, everybody's going to make a lot, and it's going to be saturated very quickly. So you see that by multiplying the readout, by multiplying the type of samples that we have in terms of donors, we're able to explore kind of all the possibilities in terms of dynamics, in terms of the way this inflammation can be set up in this ex vivo system. And again, what's very striking here is that these were 6 healthy donors. As far as we can tell, there is no obvious differences between them. No, the age is more or less the same. They are different sexes, but no, again, well balanced. And yet, these healthy donors make CAR T cells, which makes a lot of IL-2. And these healthy donors in purple doesn't make any IL-2. And vice versa, it makes a lot of MIG and not so much MIG for the brown and so on. So really, when you think about it, now we are used to doing a lot of immunology in mouse. And when we move to human immunology, we need to have new tools to be able to really explore the diversity of the response. Of course, we cannot do as much in vivo, but maybe by building these robots, by being able to explore the space of all the possibilities of these cells doing different things, depending on the type of APC, depending on the type of tissue, we might have a better sense of the way T cells make decisions and maybe build a better model of the way T cells get activity inside tissue. Now, the challenge in terms of modeling is that when you look at the data and you collect, again, here I'm showing you different clusters of T cells measured by spectral flow cytometry and different level of cytokines. For different time point in a gray, different quantity of antigens in orange, yellow, and different quality of peptides or antigens in green and yellow. And you see that there is no clear pattern. It's not easy across different time points. It's not easy to distinguish between, let's say, a good antigen in yellow and a bad antigen in greens, depending on the time point, depending on the quantity, depending on the context. You could have exactly the same amount of cytokines, you could have exactly the same kind of T cells. But at the same time, we know that the immune system is kind of very clever, is able to deconvolve this stuff. So how can we take these big matrices of data, right, this big dump of different level of cytokines, different type of activation of T cells, and try to build a very low dimensional model, a very simple representation of the way T cells get activity in such a way that we can go back to the mouse, we can go back to our patients and really understand the way this immune system gets activated. So that brings me to the third part of the talk, which is really to develop some machine learning tool to really build a model of immune responses, in particular to build a model of T cell activation. And the analogy that I always make, because I think it's very striking and people really understand it well, is that at some level, we immunologists living before 50, and I'm speaking of European astronomers, we really believe that the Earth was the center of the universe, right? So before Copernicus, before a good model of the solar system, the Greeks, the Arab astronomers, European astronomers were building models. I have to say, I don't remember when the Chinese astronomers figured it out. I think they figured it out before the European, but I'm sorry, I'm going to give this analogy from the Copernicus point of view. So again, if you have a model of the way your system works, if you believe that the Earth is the center of the universe, you get very complicated dynamics and it's very difficult to understand what's going on. And of course, if you have the right idea and if you have the right type of model where you put the sun in the middle of the universe, if you are Copernicus, suddenly things start to make sense, right? You get these very nice trajectories. They are very regular. You have a better chance to go to the moon if you know the laws of physics from the right compared to the left, right? So that's really where we are at, I think, in terms of immunology. We have a lot of observation where we observe a different type of T cells, different type of cytokines happening in different tissues. But we kind of have a very hard time of organizing the data to really understand, to predict what's going to happen if we inject different vaccines or if we perturb the system in different ways. So we really want to get to a better set of variables which are really going to help us drive the immune system in a better way. So it turns out that people in machine learning have been developing some tools to do that automatically. So this is an example, again, taken from astrophysics, where you can build some neuronal net called an autoencoder. It takes your data, right? It takes this observation as kind of the angles between all the different planets, right? So take it as a proof of principle where if you just measure all the angles between Mars and the Earth and Jupiter and the Sun and feed them inside this autoencoder, what you're going to learn is that the best way to squeeze the data in low-dimensional space is really to measure all the angles from the sun, right? So automatically, from a bunch of observation, you're going to learn that the best way to represent the system is really to learn that the sun is at the middle of the solar system, and you really want to measure all the angles. And suddenly, all the trajectories in the latent space, right, in these latent variables, look very simple and very regular. So I give this example just to show you that people do that in physics and in machine learning. And we are trying to do that in the context of immunology. And I hope you are interested in this kind of approaches, because I think there's a very good discussion to be had and a very good system to develop machine learning. So here on the left, I'm showing you the raw data, right? I cannot show you a 60-dimensional space with all the different cytokines and all the different type of T cells developing inside our robot. But just in 3D, you see already that things are a bit complicated, right? You know, simply measuring IL-2, interferon gamma, and TNF on the z-axis for different quality of antigens in the OT1 training set. You see that the blue is mixed with the orange, and it's very non-linear and difficult to disentangle. So it's a bit of a mess, if you ask me, to really build a model of this stuff. Now, the first insight that we have is that if you, instead of looking at the cytokine itself, you look at the time integral of the log of the cytokine. So you are kind of looking at the cumulative inflammation inside these dishes. You see that things start to be a bit better organized, right? The blue starts to be separate from the pink and so on and so forth. So just taking the data and starting to modify the variable in a better way. So in this particular case, time integrating the cytokine is really a better way to look at inflammation, and we can discuss how relevant it is to most immune systems, but it's really the way we look at the inflammation most of the time now. And now you can take this data. So you start from the raw data in the robot, you compute the time integral of the log, and then you feed that into a neuronal net. So again, I'm just showing you a very simple version with 5 cytokines. And what this neuronal net does is that it takes this very high dimensional space and try to squeeze it into a 1D or 2D, one-dimensional or two-dimensional space, so kind of very compact representation of these very complex dynamics. And we build this space in such a way that it's going to really distinguish the different antigens just based on their sequence, right? We're asking, we have all these different patterns of cytokines. We have all these different sequences of antigens which correspond to different type of bug in a way. Can you build a low-dimensional space where these things are going to be well organized and you're going to be able to classify the response in a very nice way? And indeed, when we have this geometry and these different weights, this is what the latent space, so this kind of a low-dimensional space looks like. Now, we started again with a very complex 60-dimensional, very dynamic and nonlinear cytokines and cell differentiation. And now, when we look at the latent space, things are very simple and very simple dynamics inside the system. Not only that, but this is really an important thing, which is that this low-dimensional space is all you need to know about the system. We built it from the cytokines, but once you have that, you can go back and build the cytokines back, right? So if you give me some data and I build you the latent space and I transmit you this picture or this dynamics inside the latent space, that's all you need to know to be able to rebuild all the cytokines which are being produced by this population of T cells in all these different inflammatory contexts. So this is really a compact representation, a very good compression of the data, a good model of what's happening inside the system. And now we can try to understand the immunology by looking at this space and this very simple representation. So for example, on the right, I'm showing you that like the model that we have on the left, on the y-axis, is completely, can rebuild all the data, right? We have all kind of different cytokines, all kind of different time points, all quality of antigens across almost like 4 decades. Different. Some cytokines go like from like 100 femtomolar to 10 picomolar. Some cytokines go from 1 picomolar to 1 nanomolar. it doesn't matter. We can really capture everything simply by compressing the data in this latent space, and we know everything about the system. So if you give me that, I will be able to tell you how much cytokine was inside your mouse or inside your patients automatically. Not only that, but because it's a low-dimensional space, now we can start doing some math. So we can take this time trajectory for T cells getting activated, again, and parameterize it. And the beauty is that it might be very complicated dynamics. We have very complicated equation. Maybe 4 parameters are necessary to really understand the dynamics. So if you want to think in terms of immunology, there is a speed, which is the way T cells are really getting activated, how fast they start producing their cytokine. There is an angle in their trajectory, which is really the balance between innate cytokines and adaptive cytokines. There is a time scale, which is really how long the T cells are being activated. And there's the long-term dynamics, which is really the way cytokines get consumed. So at some level, we can parameterize the immune response with these 4 parameters. But when we look at our data, we notice that these 4 parameters are all proportional. So they are practically, if you give me the immune speed, I can tell you what's the balance between adaptive and innate. I can tell you how long these cells are activated. All the information is going to be encoded inside that single parameter, which is really the immune speed for the system. So we started with a very complex set of cytokines and T cell activation. And now I'm left with a single parameter, this immune speed, which tells me everything that I need to know if I really want to be able to classify the immune response according to the antigens, to really be able to feed the data and really be grasp the complexity of inflammation inside the system. So I'm going to skip this part for the sake of time. I can do a bit more math. Let's go to some applications. So again, we start with simple T cells or simple mouse TCR transgenic 31 T cells. We have a training set based on a few peptides. We can test different quantity of antigens, and we get this time dynamics inside our latent space, right? We can build this very simple low-dimensional representation of the time dynamics of T cell activation. Now, this latent space is all you need to know to be able to classify the response. So again, we can parameterize, sorry, this trajectory should come about, measure the speed at which you move and how fast you bend and so on and so forth. These are all immune parameters which relate to maybe what you care about in terms of specific cytokines, but everything can be quantified in terms of this immune velocity. And here, we are measuring immunogenicity for our training set with the circles, and we measure it as a function of the quality of antigens as measured by EC50. And if you give me a new antigen that I've never seen in my model, and I measure the V0, I'm going to be able to really tell you what the quality of the antigen is going to be. So this is extremely powerful because it means that if I don't know what peptide you are dealing with inside your tissue, right? If you don't know what type of tumors we are dealing with, we don't know what type of viral antigens we are dealing with, simply by looking at the time dynamics of what the T cells are doing, I can quantify the kind of EC50, even though I have not purified the peptide and I can't do a dose response. Yet, by measuring all these cytokines, by compressing it into a low-dimensional space, I'm going to be able to classify whether you're dealing with a hot tumor or very antigenic virus, or something which is more like the self in a very quantitative manner, in a very robust manner. So this is kind of a validation set that we did. Now, we did that, again, with a very simple mouse T1 system with splenocytes pulsed with antigens. But you can use different type of antigen-presenting cells, like tumors. So this was more like a vaccine. We pulse the antigens on the B cells. This is more like a tumor where they constitutively express the antigens. We use the same neuronal net and the same projection, and yet we can still line up the data. So there's a bit of a shift because tumors are not as good as B cells activity, but a good antigens on a tumor is a good antigens on the B cells, and so on and so forth. So you can really start to learn how you can translate what you know in the context of maybe the spleen to the lung, to tumors, to a different tissue. The ranking of the antigen is going to be very consistent in terms of T cell with just a little bit of rescanning. And that way we can really compare immune response from a tissue to tissue. We can do the same with different types of antigen-presenting cells. So myeloid, for example, we strain with the splenocytes. We can do it with bone marrow-derived macrophages. We can do it with bone marrow-derived dendritic cells. The same latent space, the same model is going to apply. You're going to be able to rank the antigens again in a nice way. no matter what the type of co-stimulation or inflammation you have, the same model is going to really translate from splenocyte to macrophage to dendritic cells in a very robust manner. So this is very striking again, because if you had looked only, let's say, at interferon gamma, or if you looked at only at IL-6, this different type of tissue would give you very different response. The same peptide with splenocyte might be way weaker compared to dendritic cells. But again, because we bag together all these cytokines, and because we really model all these cytokines, try to compress all these cytokines at once, we can really build something which really tells us that T cells are able to discriminate between ligands. No matter what the context, we are still able to discriminate the good ligands in blue from a very weak ligands in red, with a little bit of a shift maybe, but again, you cannot fool of T cells. Something which is really antigenic is always going to be very antigenic, no matter what the tissue, no matter what the context. So not only that, but if you do the same trick of loading up CAR T cells, so mouse CAR T cells, so this was done in collaboration with Naomi Taylor and Yori Shah, again at MCI. So we build mouse CAR T cell which recognize CD19 on the leukemia models. And we compare the CD19, anti-CD19 CAR T signaling compared to a TCR signal. And what you see is that surprisingly, we know that these mouse CAR T cells are very good at killing, but in terms of activation, in terms of antigen response, they look like a very weak ligands. So there is this idea that, again, we can really improve on mouse CAR T cells. Yes, we can make a lot of cytokines very quickly, but it's not very sustained. It doesn't look like a very good T cells. We're really working with Naomi and Yarli to really build better CAR T cells, because we have a very good model of the way these T cells are activated. And we hope to come with better CAR designs very quickly based on this model. And I'm going to finish with more immunology. So I started by telling you that this is a very complex signaling machinery with a lot of kinases and adapters and a lot of different receptors coming about. So in collaboration with Paul Love's group at NICHD, again in Bethesda, and this is work from Guillaume Go with a postdoc in Paul Love's lab, they built a mouse which is mutated for the tyrosine on the zeta chain of the TCR, right? So you have a T cells which has the same receptors. It does have a CD3 chains associated with it, but the CD3 zeta chains are mutated and they cannot signal. So at some level, we are really asking, do we really need all these different tyrosines on the TCR? How does our model of the wild type mouse is going to look like when we start to mutate the TCR chain, associated chains in the T cells? So I miss telling you this explanation, but basically when we look at this latent space, we can build this latent space with two latent space variables. The latent space variable number one, as a function of different quality of peptide, is a simple kinetic profiling scheme. The better you are at binding, the more you activate this variable. And in a way, you are just doing kinetic profiling. And variable number two, which is a second variable in this space, has a very interesting nonlinear relationship where you're going to activate something negatively when you have a very weak peptide. So if you remember, I started by showing you our adaptive kinetic profiling model of the T cells, and I told you that you could build a good model of T cells by having a positive signal competing with a negative signal. Well, when we analyze our latent space and when we analyze the model derived from the cytokine, we recover exactly that model. One node seems to be an accelerator. The better you bind, the more you activate that node in yellow. And the second node is more like a negative feedback. When you're in an intermediate regime, you're going to activate this negative feedback, and you're going to be able to really build a good T cell by this competition. But you see that for wild type T cells. And now when we go to Guillaume's T cells, and when we measure the cytokine and we measure the latent space, what we see is that latent space number one, again, the better you bind, the more you activate this variable. It's still a kinetic profiling scheme, but a little bit shifted to the left. So you are better at responding when you mutate these tyrosines. But more interestingly, when we look at latent space variable number two, there's no more negative feedback. There's no more of this negative signal. And this signaling impaired OT1, they are missing the negative feedback. And really, they are going to be kind of deficient in the way they do self-non-self discrimination, because they are missing this negative feedback as discovered by our machine learning and by our model of T cells. So indeed, when we plot the data, if we look, for example, at just gamma, interferon gamma versus IL-2 integrity of the time, when you look at the wild type T cells you have all these classes of T cells with different level of activation. You can really separate them based on the quality of the peptide MHC T-cell interactions. And when you move to Guillaume and Paul, the mice, which are missing some of the signaling units in the TCR, it's going to be all or none. Either you are a T-cell which is completely quiet and you don't make any kind of cytokines, or you go full blast and you completely saturate the system and you miss all this intermediate step because you are missing the negative feedback, which enables you to really discriminate in a very fine manner. So so this is not published, this is still under review, but we like very much this analysis because simply by looking at the latent space, right, simply by taking the T cells from Guillaume and Paul's mice and putting them in our robot, measuring the time dynamics and building this latent space, we learned that the big defect inside this T cell was this negative feedback. And we learned that in a way, these T cells are very bad at discriminating. They are completely digital. They do all or none. Either they are fully activated or not at all. They are not able to do the fine-tuned activation that you see in the wild-type T cells when you have a full signaling machinery in place. And we built some model for that, but again, this is under review, and we hopefully will come out very, very quickly. So I'm going to conclude here to leave some time for questions. I hope I give you a sense of something I'm sure all of you are very familiar with, which is that when we look at T cell activation, when we look at any cell of the immune system and we try to measure inflammation or measure the way these cells are activated, this is really a very high dimensional space. There are a lot of readouts that we can accumulate. It's very complicated to deal with that. Now, being able to build a robotic platform to take these T cells in this case and just kind of activate them and see what they do as a function of time and what they do in a very reproducible settings, we can really build large data sets fairly cheaply and very quickly. And we can work with a lot of, with very small amount of T cells and clinical product. So in a sense, from very few cells, you can really start to kind of rev up these cells and try to understand how they respond to a stimuli. And again, I hope I give you a flavor that there are some tools being developed for machine learning, which enables you to really take these very complex data sets and really compress it into something manageable. So we can really start to understand, we don't want to look at just interferon gamma or just effector memory cells in the CD8 compartment. We want to look at all these observables and try how to see how they are related to each other and try to build a very low dimensional space to really understand how this system makes decisions. So in the particular case of what we use that to really quantify antigenicity. And again, to learn a model of immune decision, which really applies to T cells in many different contexts. It's really a competition between a positive signal and a negative signal, which really decides the way T cells get activated. Now, if you want to read a bit more, this got published 6 months ago now in Science, and there's a lot of math being done. You can skip it if you don't like it. You can go into the details, and please come back to me and ask questions if you need to understand better. But really, this is really kind of a blueprint of what we're doing in the lab at present time, because we can really build complex system in the robot and really go and probe it and model it in a quantitative manner. So we have postdoc positions available if you're interested in this kind of approach. And I'm going to finish with people who did the work. So a lot of the work from my lab came from Suraj Achar, who is a grad student. He's doing a fantastic job building the whole pipeline from the robot to the data wrangling to a lot of modeling of the data. And you can really, the jack of all trade who works very, very hard to do all these things, with some help from Madison, who is pushing the more single cell approaches. And Angela Lee started the work building the robot with me. And Emmanuel Salazar Cavazos, who is a postdoc who does a lot of quantitative studies on the tumor cells. And a lot of this theoretical approach was developed in collaboration with Paul François' group at McGill, in particular François Bourassa and Thomas Rademacher, who were grad students. And we're getting a lot of help from different people at NIH and different places across the world. Again, if you're interested in this kind of work, don't hesitate to contact me. We are a small team, very interactive. We can tell you that NIH is a very open place and very interactive place. Joanne can attest to that, and really, it's really a way to be interdisciplinary. And I hope we speak and we collaborate in the future. Thank you very much, and looking forward to your questions. And I'm going to finish with this video of all the different dynamics that we measured just to give you some inspiration. Thank you so much.